Hello, everybody. My name is Roberta Cardenas, and my capstone project was on DNA barcoding of crab megalops in West Hawaii to understand patterns in larval recruitment. For a little bit of background, larval dispersal is very important for coral reef species as it connects and replenishes their adult populations worldwide. Brachyrian crabs have a pelagic stage called the Megalops stage, and this is when they settle into their habitat. So this is very important for setting the timing of recruitment in crabs. However, they are very challenging to study using morphological methods. Here on the right, I have a figure showing different species of crabs at the Megalops stage. And as you can tell, they are very identical. But DNA barcoding allows for accurate and efficient species identification. Crabs are very important here in Hawaii as they support a variety of fisheries such as the Kona crab, also known as the Hawaiian blood-spotted swimming crab, and the mud crab. They also play a key role in benthic communities as scavengers and mid-trophic predators and prey. Currently, there are 284 brachyrian species found in Hawaii. However, there's no scientific documentation of the diversity or spatiotemporal distribution of crab larvae in Hawaiian waters. For my research question, I wanted to know what species of crab megalops are found within West Hawaii, and within that, does the density of each species vary between slick and ambient habitats? My samples were actually collected by Dr. John Whitney and his team over at NOAA. Here on the right, I have a map showing the different sites where the samples were collected. The samples were collected inside and outside of slicks, and for a bit of background on what slicks are they are smooth areas of water surrounded by rippled water. And these are very important for larval populations as they cause the larval organisms to accumulate within the slicks and can serve as nurseries. The samples were collected during 3 field expeditions ranging from 2016 to 2018, and they used nucentos to collect the plankton inside and outside of slicks. The first thing I did was I sorted through each of these jars shown on the left, and I used a dissecting microscope to sort through each individual sample and pull out each megalops that I found. And these are some of the pictures that I found while sorting through these samples. To DNA barcode these organisms, I first performed a hotshot DNA extraction, and I used that DNA to amplify it using PCR, and I used gel electrophoresis to confirm that I had amplified enough DNA that would allow for species identification. So once I had amplified that DNA, I sent it over for sequencing over at UH. And I was sent back very long sequences that I then put through a barcoding database. And this database had known sequences for different species. And I was able to compare my sequences with the sequences in that database. And I used a 95% identity threshold to match up the sequences and identify the species. Within all the megalops that I pulled out, I successfully identified 60 of them, and within this, I found 12 species across 7 families. Here I have a picture of one of the megalops that I found and what they look like as an adult. This is a rank abundance plot showing the distribution of abundance in all of the species that I found. So as you can tell, the most abundant species was the hairy swimming crab, shown all the way on the left. And next to it is the red swimming crab, which was the second most abundant crab. And as mentioned, I also found a Hawaiian blood-spotted swimming crab, which is endemic here to the islands. I also compared the density of the megalops for each year in both ambient and slake habitats. In 2018, the density of megalops was higher in ambient habitats, but overall, the density was higher in slicks across all the years. I then ran a ANOVA to find if there was a significant relationship between the years in the habitat, and as expected, I found that there was a significant relationship between the density and the habitats. In conclusion, I'm going to continue sequencing these megalops, which will allow me to increase the amount of species that I found. And by having more species and increasing the species richness, this will allow me to investigate seasonal patterns in the timing of recruitment by comparing the density of each species across the years, as well as in both ambient and slake habitats. Big thank you to my advisor, Dr. Yaki, as well as Dr. Chelsea Council, as well as my lab mates over at the OI and everybody at HPU and the OI staff. Also, Dr. John Whitney, who provided me with the samples, and the NOAA Kona Integrated Ecosystem Assessment Team.